Good afternoon. Welcome back to Virology. All right, today we are going to talk about making RNA again. Last time we did that, right? Except this time the template is DNA. It's not RNA. This is transcription and RNA processing. So here's our Baltimore scheme again. We have mRNA in the middle. And today we're going to talk about DNA viruses whose genomes are transcribed to make mRNA. And so that's transcription. And it happens from double-stranded DNA templates. It's always from a double-stranded DNA template. Remember that. It doesn't happen from a single-stranded DNA template. It doesn't happen from a gapped double-stranded template. It's always double-stranded DNA. So that is transcription, the production of mRNA from fully double-stranded DNA. So if you are single-stranded DNA or gapped DNA, you have to do something about that. You have to get the double-stranded DNA. All right, so these are the viruses we're going to talk about today, how they get to mRNA. Now here's another, I always tell you, you got to remember this, but there's certain things that are key to understanding, like, can double-stranded RNA be translated? Nope. Nope. But every year, someone gets it wrong. It's so intuitive, right? Double-stranded is all wrapped up. Anyway, here's another one. In cells infected with DNA viruses, at least one protein, sometimes many, are needed for DNA replication. So what does that mean? When a DNA comes into a cell, before it can replicate, it needs to make proteins. It means to make mRNA, and they have to be translated. But the problem is that I've just alluded to, not all DNA templates are ready for transcription. You have to be double-stranded DNA in order to be transcribed, okay? And so here's a virus, SV40, which we'll talk a lot about, where it's a double-stranded circular DNA virus. So there's the DNA, double-stranded circular DNA. And amazingly, in the virus particle, the DNA is what we call chromatinized. It's wrapped around histones. This is very rare for viruses. So when this DNA gets in the nucleus, it is ready for transcription. It's double-stranded DNA. So there are some viruses whose templates are ready for transcription, but not all. So there are three viral DNAs that have to be converted to templates for transcription. We're going to talk about two of them today briefly. So we have the Adenoviruses, where the DNA is circular, double-stranded, but not fully double-stranded, right? It's got gaps. It's also got a protein stuck on the end. It's got an RNA. Why that is, you'll find out later. It's all a consequence of replication. But that cannot be transcribed. It cannot be made into mRNA. The enzyme won't recognize this. So what happens is, this is very interesting. When broken DNAs come into the nucleus, the cell fixes it, because the cell does not like broken DNAs. So broken DNAs include gapped ones like the Herpesvirus genome, and also single-stranded DNAs of the parvoviruses. It comes in the nucleus, the cell sees it, and there's what's called a DNA repair response, and it makes it double-stranded here. This red business is added by the cell. It is not genome replication. Genome replication is making many copies of a DNA from a single one. That's replication. This is just fixing the one DNA that comes in, and now that can be just transcribed. Same with the Hep B. It's fixed. The cell fills in the gaps, takes off the RNA, takes off the protein. It's now a beautiful double-stranded DNA that can be transcribed. And retroviruses, we're not going to talk about them now, but they are RNA genomes that are converted to DNA by reverse transcription, but they're not ready for transcription yet. They have to integrate into the cellular DNA. And then they are a transcription template. So even they are not ready for transcription, and we will talk about those later. So what templates are ready for transcription? Well, the ones that are double-stranded DNA, like adenoviruses, herpesviruses, and the polyoma and papillomaviruses. These are the polyomaviruses I alluded to here. They have double-stranded DNA, gets in the nucleus, ready to be transcribed, and the papillomas as well. So there are some that are ready to go, but others have to be repaired. And this is all because you have to make at least one protein before you replicate your DNA. Now, obviously, DNA replication is essential, right? Virus gets in a cell. What's the point? To replicate, to make more viruses. And so that involves DNA replication. To get DNA replication, you need at least one, sometimes many proteins made. So what are the enzymes here? Last time I told you there are 4 kinds of nucleic acid polymerase, right? There are RNA-dependent RNA polymerases. There are DNA-dependent DNA polymerases. There are DNA-dependent RNA polymerases, which we're going to talk about today. And then there are RNA-dependent DNA polymerases. So these are the DNA-dependent RNA polymerases. There's several of them because they make different kinds of mRNAs, but they all look the same. And these are cellular enzymes. So most viruses we're going to talk about utilize the cellular DNA-dependent RNA polymerase. And there are 3 of them, Pol1, Pol2, and Pol3. You may have studied them in biology before. But Pol2 is the one that makes messenger RNA precursors for the cell and lots of other short RNAs. And some viruses, obviously many viruses, utilize those to make their messenger RNAs. There's also Pol1, which makes the precursor to ribosomal RNA. As far as we know, no virus uses that. And then Pol3, the precursor to tRNAs. So everything is specialized in the cell. We have RNA, ribosomal RNA, mRNA, and tRNA. We have a polymerase for each. Okay? Some viruses encode their own DNA-dependent RNA polymerase. Those are the ones that reproduce in the cytoplasm. Because where is the DNA-dependent RNA polymerase? It's in the nucleus. And if you don't go in the nucleus, you have no access to it, so you have to make your own. And it's the giant viruses and the pox viruses. They reproduce in the cytoplasm as opposed to the nucleus, so they need to encode their own RNA polymerase. Now you may be asking, why? What's the function of this? What is the function of some viruses going in the nucleus and some not? There's really no answer except that it works, it's evolutionarily selected for. You just have to remember that if you're going to replicate in the cytoplasm, you need to encode an RNA polymerase because they exist only in the nucleus. So here's the process of transcription outlined for you. We start with DNA template. We're in the nucleus. So here's the nuclear membrane, and here's the DNA template. We have the initiation of transcription at what we call promoters. We usually designate those by a red arrow. That's where transcription begins. And the transcript is typically capped at the 5' end. It's an unusual structure. We'll talk about it in a few minutes. And then you have a pre-mRNA made, which has exons and introns. And the introns are going to be removed, intervening sequences, and the exons are going to be in the final mRNA that's exported. And so in the nucleus, you have splicing occurring to remove the introns. And here now we have an mRNA capped without the intron, and it's polyadenylated. Polyadenylation is another step that happens in the nucleus. And only then can it be exported into the cytoplasm. And of course, that's where it's translated, right? mRNAs are translated in the cytoplasm. And mRNAs don't last forever. They have a half-life. Minutes, hours, even seconds, depending on their function. So eventually they are degraded by specific systems. Now, as we'll see, splicing is actually required for nuclear export of mRNAs. That's shown on this slide here. This mRNA is fully spliced. It's needed for export. But there are some viral genomes and cellular mRNAs that are not spliced, and they get around it. And we're going to talk about how that happens. Transcription, the production of mRNA from DNA is highly regulated because you don't need to be cranking out maximal amounts of mRNA all the time from every gene in the cell. Some you need less than others. And so the DNA is full of regulatory sequences. And that's shown here. This is a double-stranded DNA. And all kinds of regulatory sequences that impact transcription. This is called the transcriptional control region. And it consists of the initiator sequence where transcription begins, and that's our arrow, our red arrow there. That's where the mRNA would start to be made. And then we have what's called the TATA sequence, which binds a specific protein needed for initiation. And that constitutes what we call the core promoter. Then there are upstream, there are regulatory sequences, local ones, distal ones. You can see these are 100 to 500 bases away, and sometimes thousands of bases away. So all of these are involved in controlling transcription because, as I said, it needs to be regulated. And this is the biggest regulation step in after transcription, or in the whole process. Transcription, initiation, elongation, termination. It's initiation that is the most regulated. So here's an example of 3 different viral genomes and their transcriptional control regions. They're all very different because they infect different kinds of cells in you, and you need different amounts of the different proteins. So here's adenovirus, the major late. Adenovirus has multiple promoters, major late promoter, 1 initiation site, and a couple of upstream sequences for binding regulatory proteins. So regulatory proteins bind these sequences and regulate transcription. We're not going to go into those in very much detail, but they are present in order to regulate the extent of transcription. Here's SV40 early promoter. This one has 3 start sites and a bunch of upstream regulatory sequences. These are all SPL repeated over and over and over. And here's another adenovirus promoter. Many of these DNA viruses have multiple promoters, and this one has, as you can see, start sites, 4 start sites, and then E2F and ATF control sequences. All right, so surrounding every initiation site, there are these regulatory sequences. So in the DNA, those sequences bind proteins, regulatory proteins that participate in regulating transcription. So SPL is a sequence that binds a regulatory protein called SPL. So here's the process of transcription. You're never going to be asked anything about this, as far as I know. I just want you to see it so you can conceptualize what we're talking about. So this is the double-stranded DNA on the top. There's your promoter. And look at all these proteins that have to assemble at the promoter in order to get mRNA made. You have the polymerase, but not just that. You have lots of other proteins, initiation proteins, including proteins that are binding those regulatory sequences. Once they all bind the promoter, you have what's called a closed initiation complex. It's closed because it can't do anything. It needs energy to open it up in the form of ATP, and now it's open, and it can start copying the DNA and making mRNA. These polymerases make a lot of short transcripts, and then finally they get started. So you can think of it as they go 20, 30 bases and then stop, and the product falls off, and then they start again and again. And we think that's because these are all primer-independent enzymes. If there were a primer, we think it would be easier for these enzymes to start making mRNAs. But there's no primer, so you get these abortive transcripts. And finally, you then start to get transcription of the RNA. Here it is. This is the mRNA. The polymerase moves down the DNA and makes mRNA. So there on the right is the polymerase. Which looks like the polymerase we talked about last time, the poliovirus polymerase. There's an active site, and there's 2 magnesiums, 2 metals that you need, right? There's a channel in the polymerase so that the NTPs can come in. Right? We're gonna make RNA, so these are NTPs, not dNTPs. And then we have the double-stranded template coming in here. It's denatured within the enzyme. You see that these structural features of the enzymes are actually separating the 2 strands. And then the single strand goes through the active site, and the active site makes mRNA, which is colored red here. And then it goes out the mRNA exit channel, and then the DNA refolds and becomes double-stranded. So as the polymerase is moving along, this is what's happening. The DNA is being denatured, it's transcribed, and then the double-stranded DNA comes out. You may ask, do these enzymes make mistakes? Yeah. It doesn't matter as much because mRNAs are not the genetic material. These are just for making proteins. So if you make a few proteins with the wrong amino acid, it doesn't matter. It's not going to be passed on to your offspring. If this were a genome and you made a mistake, then the mistake would be passed to the offspring. But the mistakes made during transcription really have minimal impact. So there are some very long-distance regulatory sequences called enhancers. And this is how they work. So here's a DNA, and there is your initiation complex. There's your RNA polymerase 2 and all these other proteins that need to bind, and there's your transcription initiation site. There are distal sequences that can be up to 10,000 base pairs away. They're called enhancers because they enhance initiation. How do they do it? There are enhancer binding proteins that bind the enhancer sequence and also this initiation complex and stabilize it and make it initiate better. There are also sequences that downregulate transcription at a distance as well. So this is an unusual feature of eukaryotic enzymes. So we mentioned proteins that regulate transcription by binding to the DNA. These can be host or virus, and we'll see some examples of these. They are sequence-specific DNA binding proteins, right? So I showed you these protein binding sites with weird names. These are sequence-specific. In other words, SPL protein will only bind the SPL DNA sequence and not USF, and the same for USF and so forth. So they are sequence-specific, but there are also regulatory proteins that don't bind DNA. They're called co-activating molecules, but they can regulate transcription. And the way they do it is by modulating the structure of the nucleosome. We'll see an example of that in a minute, but as you know, DNA is wrapped around histones, and the structures are called nucleosomes. And how tight the DNA is wrapped around the nucleosome determines how well it's transcribed. If it's really tightly wrapped, transcription is poor. It's a way of regulating transcription. If it's loosely wrapped, transcription can go up. So how do you control the wrapping? By histone methylation or acetylation. So you can make chemical modifications of the histones, and that either loosens the DNA or tightens it up. So these proteins that do this are transcriptional regulators, but they do not bind DNA directly. They simply modulate histones. We'll see an example of that in a bit. The sequence-specific transcriptional regulatory proteins have a modular organization like this. So they have this is the protein here from N to C terminus, has a couple of different domains, has a nuclear localization signal. It's a sequence in the protein that puts this protein in the nucleus. That's where you need it, right? Because transcription is happening in the nucleus. So these are made in the cytosol and shipped into the nucleus by the NLS. They often act as dimers, and so there's a region needed for dimer formation. Typically what's called a leucine zipper allows dimer formation. There is a DNA binding domain, which allows the protein to bind to the DNA, and specifically so that sequence determines the binding. And the motifs involved in DNA binding are called zinc fingers, helix, turn helix, basic sequences. They're all involved in DNA binding. And finally, at the C terminus is the activation domain. So the protein binds to the DNA as a dimer, and then it has to activate transcription by binding to the transcriptional complex, perhaps, and that's what that region is all about. But these sequences differ, and that's what gives these proteins specificity. First question, what is the first biosynthetic event that occurs in cells infected with double-stranded DNA viruses? Biosynthetic event. Membrane fusion, transcription, DNA replication, protein synthesis, all of the above. Now, so what I mean by biosynthetic is to make something, make a molecule. Okay, how did we do? Yep, transcription, right? You need to make one, at least one, sometimes many proteins. So to do that, you need to make transcription. It's not DNA replication. To get DNA replication, you need to make proteins. So that means transcription and then protein synthesis. Not all of the above either. Okay, now an overview of transcription of different kinds of viruses. There are some viruses that utilize only the host transcriptional system, which includes the polymerase and accessory proteins. And these include some retroviruses, some plant viruses, cauliflower viruses, yeah, and circoviruses, those little single-stranded DNA viruses that are in all of us. Okay, they only use the host system. Then there's some viruses that use the host plus one viral protein. So they make one viral protein that gets involved in transcription, and that includes the bacteriophages T3 and T7, which make a protein that is involved in transcription of the late genes. And then these viruses here make a viral protein that regulates transcription. Adenoviruses, papillomas, polyomas, retroviruses, et cetera. All right, and then finally there's some, well, not finally, penultimate host plus more than one viral protein that stimulate transcription. So adenos and herpes make more than one viral protein that get involved in transcription. We'll see example of that. And there's some where the whole transcriptional system is viral, right? Those are the ones that replicate in the cytoplasm because they have to encode the host system because otherwise it's in the nucleus and they can't access it. Now, a key thing here is that if a virus is going to be transcribed only by the host, it needs to recognize viral promoters. The host needs to recognize viral promoters. So for these viruses, these promoters have to be recognized by the host. In some cases, there is a viral protein made that allows the host to recognize viral promoters, and that includes the herpes viruses. So there are some general strategies of transcriptional regulation that we're going to talk about, and they're shown here. They're called cascade regulation and positive autoregulatory loops. So these are examples of how viral proteins can regulate transcription. So a virus gets into a cell, it's using mainly the cellular transcription machinery, but it can make proteins that modulate it. And these are ways that it does that. So at the top, we have cascade regulation. So we have a viral genome coming in the cell. And we've got two genes here, X and Y, each with their own promoter. And gene X is transcribed by cellular proteins. It's made into an mRNA. And that mRNA is made into protein X. Protein X is needed for transcription of gene Y. Gene Y will not be transcribed unless protein X is made. So it's a way of regulating gene Y. Maybe you don't need it till later, and so you have it dependent on protein X, and then that is transcribed to make mRNAs in the protein. So that's cascade regulation. And sometimes it gets pretty extensive. You can have X doing Y, and then Y activating another gene as well. So if you need to regulate transcription, this is one way to do it. And then on the bottom, we're showing a positive autoregulatory loop. So we have a genome with a single gene and then a promoter. It is transcribed by cellular proteins, as was the case for A. Transcribed by cellular proteins. Viral RNA is made, protein X is made, and then protein X goes to the promoter and enhances the transcription of gene X at the promoter. So it's positively autoregulating transcription. And so you get more of the viral mRNAs made once protein X is made. Another way of regulating transcriptional activity. This can also be negative autoregulatory. It can be that you make gene X initially, and then after a certain amount, it shuts itself off because you don't need it anymore. So it could be positive or negative autoregulatory loops. So let's look at a couple of examples of this. SV40, it's the simplest one. We have a double-stranded circular DNA template. The ORI is the origin of replication. That's where DNA synthesis occurs. We'll talk about that next time. And there are just two promoters. There's an early and a late. Those green arrows show you the transcription, and they're going in opposite directions from the origin of replication. So early and late promoters. So right away we're seeing there's some temporal regulation of transcription going on. So what happens is when the genome gets into the cell, it's double-stranded. It is ready to be transcribed, right? Nothing need to be done to it. And this line at the top here shows the timeline of production of proteins. The early promoter is on when the viral genome gets in the cell. It encodes an mRNA for a protein called large T, or just T antigen. T antigen has many functions, as you will see, one of which is to turn on DNA synthesis. And so when enough T antigen is made, it's going to turn on viral DNA synthesis, and that is in turn going to turn on the late promoter so that you can get the late proteins made. And I think this is diagrammed. All right, so it's later. So the point here is we have an early phase where DNA replication is occurring, and then a late phase where we're making structural proteins. The late proteins are structural proteins. You don't need to make structural proteins until you have DNA to put inside them. That's the whole purpose of this regulation. You make large T that enables DNA replication, and then you make the capsid proteins. So let's look at how L is kept off early. Why isn't L on early in infection? Only the early promoter is on. So it is off because of cellular repressors. So let's look first at the graph on the left here. We have just relative concentration on the y-axis, and then the time after infection. So we have viral genomes, so that's in blue. So you can see there's a period when there are no viral genomes made, and then at some point they start to be made, so there's DNA replication. And when DNA replication begins, so does the synthesis of viral late mRNAs encoding the structural proteins, the capsid proteins. But this mRNA is off. You can see early in infection. During this period, the early promoter is on, and it's making T antigens so that DNA can get replicated. So what keeps this off early in infection? Well, it's a cellular protein called initiator binding protein, IBP. It's a cell protein. It exists at a fixed quantity in the infected cell. So you see it's here, it's shown in orange. It's the same level throughout infection. It doesn't go up, it doesn't go down. So what happens is when the viral genomes first get in, there's a fixed amount of initiator binding protein, and it binds up all of the regulatory sequences needed for transcription of the late promoter. And it's only when the genomes begin to replicate that there is eventually not enough IBP to block all the transcription regulatory sites. So you see there's initially 3, and then 1, and then none because there isn't enough IBP. Now you have lots of genomes present, and you've essentially titrated out all of the IBP. So that is what activates the late promoter. So now you have late transcription going on, encoding for capsid proteins, and then you can build new virus particles. So it's very interesting that it's a cellular protein whose concentration is level, which has the ability to bind the regulatory sequences needed for transcription of the late promoter. So that inhibits the late promoter until there's a lot of DNA made, and then there's not enough IBP to go around, so you're going to get transcription of the late promoter. That's how you ensure that that the late promoter is not on early in infection. So as I said, and this emphasizes it, the function of early and late phases is to delay synthesis of structural proteins until DNA has been replicated. This is, in my view, this is a human view of it. It's justifying it in a human-centered fashion. Why make structural proteins if there's no DNA to go in them? It makes sense to us, but I'm not sure if that's really the reason, or maybe there's something else we don't know about. But that's the way we think it's happening. So you have an early phase, the genome gets into the cell, it goes in the nucleus, you make the early mRNA, which encodes T antigen. T antigen goes in the nucleus and stimulates DNA replication. And when DNA replicates, late promoter is activated, and that makes the late mRNAs that go out into the cytoplasm and make the structural proteins. And then those structural proteins go back in the nucleus and they encapsulate the DNA that has been replicated. So it's a nice example of how transcription is regulated and for a specific purpose. Now, getting a little more complicated, adenovirus, a longer genome, linear double-stranded DNA. And there are 3 viral proteins and DNA synthesis that govern the regulation in a way that we've been talking about. So when the genome first gets in, it is transcribed. The immediate early promoter is transcribed here. And one of the products is E1A protein. This protein that looks like an eggplant there. And E1A is necessary for transcription of the early genes. So the early genes don't get transcribed unless E1A, an immediate early protein, is made. And then the E proteins include proteins needed for DNA synthesis, like E2. So DNA synthesis begins, and then that activates the late promoter, much like we saw for SV40. And there are some other viral proteins, 4A2 and L4, that also stimulate activity of the late promoter. So a little more complicated regulation in that we have 3 proteins as opposed to 1 for SV40, the T antigen. So let's see how this works. So here's our viral DNA coming in. The transcription of the early genes, not the immediate early, but the early genes requires E1A. And that's because E1A frees up a set of transcription proteins called E2F. Now, normally, in the uninfected cell, E2F is bound by a protein called retinoblastoma protein. And when this binds promoters, E2F promoters, like this, the RB component will recruit histone deacetylases to the promoter. And histone deacetylases take off acetyl groups from histones and make the DNA, make the chromatin more compact, turning off transcription. So when RB is present on these E2F proteins, transcription is shut off. And that's shown here. Here's closed chromatin because it's acetylated in this case. Sorry, deacetylated. If you acetylate it by a histone acetyltransferase, you open up the chromatin, you relax it, it becomes transcribed. If you take off the acetyl groups with a histone deacetylase, then the DNA wraps more tightly and it can't be transcribed. So histone deacetylases remove acetyl groups, wrapping the DNA more tightly and reducing transcription. So that's why you need E1A, because E1A binds RB and prevents it from hitting promoters, and not just a cellular promoter, but the viral early region promoter. It needs E2F, and it can't get it. There's E2F. It can't get it unless E1A binds RB. So E2F is required for the early region, And E1A, this is the mechanism by which E1A makes it available. Now remember this, because when we talk about, these viruses can cause tumors, these adenoviruses, and this is going to be a major player, E1A, in that process. So E1A is made, it frees up E2F by this mechanism, and then the early region can be made that makes the proteins needed for DNA synthesis. Now the DNA genome has a complicated transcriptional unit. Here's the double-stranded DNA, and both strands are transcribed. And so you have early region proteins, mRNAs transcribed, and you have late region protein transcribed. And so, in fact, in immediate early and early, as well as late proteins. And so these include, on this strand, the DNA binding protein, the DNA polymerase, everything you need to replicate DNA. And then on the other strand, mRNAs encoding the structural proteins, hexon, penton base. Those are late proteins, as you can see by the L. You don't need them right away for the same reason we discussed in SV40. You don't need them until you have DNA, and you don't have DNA until you make the early region proteins. So here's how this happens in a cell. We have immediate early, and late phases. Virus is internalized, the DNA gets in the nucleus. The immediate early mRNA is made. Only that one goes out into the cytoplasm. The protein is made, it goes back in the nucleus. It binds RB, frees up E2F, and now you can get transcription of the early mRNAs. And those mRNAs go out in the cytosol and they make the DNA polymerase. So they can go back in the nucleus and replicate the DNA. And then once the DNA starts replicating, it turns on the late promoter, and that gives you the late mRNAs encoding structural proteins, which go back in the nucleus to make virus particles. So 3 steps of regulation, a little more complicated than SV40 because of that immediate early protein that's needed. Okay, one more example, herpesvirus, to make it even more complicated, but this introduces an interesting twist. So we have immediate early, and late genes, just like adenovirus. The difference is the immediate early promoter cannot be recognized by the cell transcriptional apparatus. It can be by adenovirus. So the adenovirus immediate early promoter can be recognized by cellular proteins, but the herpesvirus immediate early protein cannot. So what do? Well, the solution is within the virus particle, there's a protein called VP16. It's a protein physically carried in the particle into the cell, and that allows the immediate early promoter to be recognized by the cellular apparatus. If not for VP16, that promoter would not work. So it's the solution to the problem. So then you have immediate early transcription. You have a protein ICP0 that then goes on to activate the early promoter. That makes mRNAs encoding replication proteins. That stimulates DNA replication and turns on the late mRNAs, which encode the structural protein. So very much like SV40 and adenovirus, it's just that the details are different. And again, the regulation ensures coordinated production of DNA genomes and structural proteins. So here's how it happens. Immediate early, and late phases, the virus infects the cell. So now herpesvirus is enveloped, binds to receptors, and it fuses at the plasma membrane. Then the nucleocapsid gets into the cytosol. It's transported to the nuclear pore by microtubule motors. Remember, there's a portal on the herpes capsid. It docks on the nuclear pore, so the viral DNA comes out of that. It's circularized, and then the immediate early promoter is active. You see these little dots here. That's VP16, which is present in the area around the nucleocapsid. It comes in and diffuses into the nucleus, and it's available to activate the immediate early transcription unit. That immediate early unit encodes a protein that activates the early gene. It's ICP0. The early genes are transcribed. They produce proteins that replicate the DNA. And then once DNA synthesis begins, the late promoter is activated. You get late mRNAs made, making structural proteins that go in the nucleus and help make new virus particles. So really similar to adenovirus, but this twist of having VP16 in the particle because the promoter is not recognized by the host cell apparatus. Next question is adenovirus E1A protein stimulating the expression of adenovirus E2, which then stimulates the expression of 4A2 and L4 late proteins, is an example of a negative autoregulatory loop, repression of gene expression, cascade regulation, dimerization. All right, let's see how we did. Cascade regulation, that's right, 94%. So negative autoregulatory, nobody picked, which is good, it's not. Repression is not, it's stimulating expression. Stimulating expression, it's not repression. So cascade regulation, that's it. Okay, looks like you got that. All right, now let's look at, for the remaining time, the mRNA that's made by this process of transcription. So on the left, we have the whole transcription process that we just talked about. And here is the mature mRNA in the cytoplasm. At the 5' end, it has a cap structure. I've mentioned cap structures multiple times. So what is a cap structure? It's nothing more than a GTP linked to the first base in the mRNA by a 5 to 5' linkage. And there are 3 phosphates in there. Now, normally, the bases are connected by 5 to 3' phosphate linkages, phosphodiester bonds. There's 1 phosphate in between the 2 sugars, right? But this one has 3, and it's a 5 to 5' linkage. So it's weird, and it's called a cap. In addition to this weirdness, there are also methylations of the cap G base and the first 2 bases as well. And the cap structure is needed for mRNA stability, getting the mRNA out of the nucleus, and many other properties as well. Cap is put on during transcription, so it's co-transcriptional capping. We have, here's our RNA polymerase. It's initiated transcription on the DNA. It's making some mRNA here. After about 20 to 30 bases, a capping enzyme comes in. It phosphorylates the polymerase, and that stimulates the addition of the cap structure. So it's added only after it looks like that RNA is going to keep going. Remember, I told you there's a lot of abortive transcripts. You don't want to be capping all those, so you wait until it's a certain length, and then you have the cap put on. Right, so that's the cap, and also what happens in the nucleus is polyadenylation. And for cellular mRNAs, we make a precursor which have a poly-A addition site internally. It's not right at the 3' end, but it's internally. So this precursor has to be cleaved, and that cleave point then is polyadenylated to form the final structure, which is exported. And the details are shown on the right here, just to show you what happens. We have a poly-A addition site, AAU, AAA, and the mRNA keeps going. This pre-RNA keeps going. All this stuff is going to be cut off and thrown away. We don't need it. This polyadenylation site is cut by nucleases, and then poly-A is added to it. Get about 200 bases of poly-A. So poly-A addition site is cut, poly-A is added. So it's different from the poly-A that we talked about yesterday. This is a summary of different ways you can add poly-A to viral mRNAs. So you can have cleavage of pre-mRNA followed by polyadenylation, which we just talked about. This is done by cellular enzymes, and all those viruses utilize this pathway. And yesterday we talked about polyadenylation during mRNA synthesis. So for flu and VSV, remember, reiterative copying at stretches of U. Polymerase encounters this U and it begins to stutter and add A's. That's the mechanism of polyadenylation. Or for poliovirus and alpha viruses, the poly A is encoded in the negative strand as a stretch of U. So there's a stretch of U in the minus strand, it's copied to make poly A, and then the poly A is copied to make a stretch of U in the minus strand. So three distinct mechanisms for adding poly A to viral RNAs. An important discovery made in virus-infected cells many years ago, 1990s, was the finding that mRNA is spliced. And that is, when it's first made, those precursor mRNAs that are made in cells, they have extra sequences called introns that are removed by splicing. Nobody had any idea that this was happening, but they were studying adenovirus infected cells, and they found that the pre-mRNA in the nucleus was always bigger than the actual mRNA in the cytoplasm. So they said, what's going on here? So they did a very clever experiment where they took adenovirus DNA, they made it single-stranded. Remember, the DNA genome is double-stranded, so you can heat this and separate the strands. And then they hybridized it to one of the messenger RNAs, this one encoding the hexon protein. And then they looked at the result in the electron microscope, and here's the actual finding. They saw all these loops. Here it's drawn so it's simplified. These bits of DNA, 1, 2, 3, that are not present in the message. They're cut out by splicing. And so here's what the primary transcript looks like. There are intervening sequences, blue, orange, and red, that correspond to these loops. These are removed. And so the actual hexon mRNA coding region is the green, small part of the mRNA. So this was discovered in adenovirus infected cells. Sharp and Roberts got a Nobel Prize for that. But now we know it happens everywhere. It happens in many, many viruses and, of course, in cells as well. And so people have studied this over the years and found out some amazing things about the process. So here is a sequence of an intron exon junction. So here's a pre-mRNA, here's exon 1, here's the intron, here's exon 2, and here's the 5' splice and the 3' splice site. So this red is going to be removed by splicing. Turns out that bases at the junctions are highly conserved. Frequency of occurrence, 100% for this G, 100% for this A, and 100% for this G. If you look at all the pre-mRNAs in the world, those bases are conserved. And that's because they're involved in splicing. And that's shown at the bottom. So here we have the pre-mRNA unspliced. This A residue catalyzes a nucleophilic attack on this phosphate, breaks the bond. So now we essentially have this phosphate is free, and we have a 3' hydroxyl on the 5' splice site. And then that hydroxyl attacks this phosphate, and now you have your spliced exons. The intron has been removed, and it can be identified as a lariat. You know, the ropes that the cowboys use, right? Lariats, because it looks like it. It's got this bond to the A and the phosphate, and there's a loop and a handle. And so this is how splicing occurs. The neat thing is, this is an RNA-driven reaction. Splicing only requires RNA, so we think it's a relic of the RNA world, which existed first on Earth. And so when these reactions occur that I've just told you, I drew them here as just RNA. But in fact, a series of proteins assembles on the pre-mRNA, lots of proteins, as you can see here. And the proteins act as the scaffold, and it's actually the mRNA that ends up causing the splicing reaction. So this is called a spliceosome, and it's a ribozyme. It's an RNA enzyme. So you don't need to know any of this except that the splicing is carried out by RNA. and the proteins are just a scaffold. Now, why do we splice? What's the function? You can get a lot of extra mRNAs made with splicing. So here, for example, are some examples. You can do constitutive splicing. So here we have a messenger RNA with 2 introns. You can just splice both of them out, and now you have an mRNA with 1, 2, 3. Or you could skip one of these exons, so you could make 1, 2, 3 and 1, 3. So that's a different protein. Sometimes there are alternative 5' splice sites. So we've talked about a 5' splice site as if there's only one, but there can be multiple. And when there are 2 different splice sites, you can get 2 different proteins made. And the same thing, you can get alternative 3' splice sites. So basically, you can expand the coding region of a viral genome. You can get way more proteins made than if you didn't do any splicing. And so that's why we think that this process evolved. Now, an example is the adenovirus genome. So here we have a 38 kb double-stranded DNA. And we're looking at just one transcription unit, the major late promoter. And that gives rise to this huge mRNA. It's almost the length of the genome. And so this undergoes poly-A addition. There are 5 poly-A addition sites, L1, 2, 3, 4, 5. So this can be cleaved and polyadenylated at any of those sites, giving you these different pre-mRNAs. And then they can undergo alternative splicing. There are all kinds of machinations that occur here. But basically, you get a lot of different proteins from splicing this precursor. You get all the structural proteins. This is the late mRNA, after all. You get all the structural proteins to build a virus particle. And beyond this expanded genome, there are examples of viral proteins that will regulate splicing. So sometimes you can have constitutive splicing, and in other situations, you can have alternative splicing regulated by viral proteins. So this really enormously expands what you can have in a viral genome. Which statement about polyadenylation of DNA virus mRNA is correct? A, it always occurs in the cytoplasm. B, it occurs after cleavage of pre-mRNA. C, poly-A is added at the 5' end of pre-mRNA. D, it is specified by a stretch of U residues in the template. Okay, let's see how we did. Look at that. A real mix. So does it occur in the cytoplasm? No, it occurs in the nucleus. I'll show you. Let's see, where would that picture be? Here. All this stuff, this cleavage, endonucleolytic cleavage, poly-A addition, that happens in the nucleus. Actually, yeah, and then what is exported is already polyadenylated. So it occurs after cleavage of pre-mRNA. Yeah, that's the right answer. So we have pre-mRNA made, it's cleaved at the poly-A addition site, and then it's polyadenylated. Poly-A is added at the 5' end. No, it's added at the 3' end, right? When you cleave here, you get a 3' hydroxyl. Anyway, the 5' end is at the other end, so we're polyadenylating here at the 3' end. And then specified by a stretch of U, many of you pick that. That's for the viruses we talked about yesterday, right? The RNA viruses get polyadenylated at stretches of U, right? Not the DNA viruses. There's a little AAU, AAA, and that gets you 200 A residues. It turns out that splicing marks mRNAs for export. What does that mean? It means if you're not spliced, you can't get out of the nucleus. Why? Well, all these proteins that bind the pre-RNA, I showed them to you in this slide here. Right? All these proteins that bind those, some of those remain on the spliced product. So here they're binding to the pre-mRNA. Splicing occurs. Now you have your RNA free of introns, but some of these splicing proteins are still bound. And those are recognized by components of the nuclear export machinery. So NFX1 and NXT1 are recognizing that this mRNA has got splicing proteins still bound to it, so it lets it through the nuclear pore. So that's a check to make sure that the mRNA has been spliced, because if it's not spliced, it's got this intron and it's going to screw up the protein sequence, right? It's going to be all messed up. So you have to make sure that it's been spliced. So this happens for most cellular mRNAs. However, there are examples of viral RNAs where the RNA has to get out unspliced. And so retroviruses, for example, they integrate their DNA into the nucleus, and then they make mRNAs. Some of them are spliced to make proteins, and they get out of the nucleus no problem. But some of them have to be full length, unspliced, because that's what the genome of retroviruses is. And so here, for example, is a retroviral genome that's unspliced. It needs to get into the cytosol to be packaged into virus particles. Now, splicing does occur, and that's no problem for those mRNAs, but mRNAs to get out. But how does the unspliced retrovirus RNA get out? Well, it turns out there's a sequence at the end called the constitutive transport element, CTE, which binds the components of the export machinery, NXT1 and NFX1, and tricks the system into exporting these unspliced mRNAs. That's neat, right? So this is not spliced, but it's got this sequence that the nuclear export machinery will recognize, and it will export it. And just to remind you, normally you get spliced, the splicing proteins remain on the spliced product, and they are recognized by NFX1 and NXT1. But in this case, the retrovirus case, we have an RNA element, basically, that is recognized by these proteins. There's always a way. HIV does it as a similar way. HIV genomes, these are retroviruses. You need an unspliced genome to get out of the nucleus to be part of the virus particle. And so for HIV, the key is actually an RNA and a protein. So there is an RNA structure at the 3' end of the full-length RNA called the Rev responsive element. This RNA is spliced, as you can imagine, to get little RNAs that make various proteins, including Rev. So splicing of this full-length RNA gives you a small RNA that encodes Rev. Rev goes back in the nucleus. It binds the Rev responsive element, and that gets it exported by the export machinery. So the export machinery recognizes Rev bound to the RNA and exports the unspliced mRNA. So two strategies for exporting unspliced mRNAs. And this actually happens for some cellular RNAs as well, but it's rare. So basically what I want you to learn from this is that splicing is value added. Alternative splicing lets you make different proteins. For a small genome, like SV40, you can make many, many proteins by alternative splicing. And you can also regulate gene expression through splicing. So it's a very flexible mechanism. And we do it too. Eukaryotic genomes do splicing all the time. It's not specifically viral. All right, one unrelated piece of information, which just doesn't go anywhere else, so it's going to go here, is RNA editing. We mentioned this last time as a way. There are ways of RNA synthesis that are not templated, right? And this is an example of that. And this happens during the replication of hepatitis delta virus. The hepatitis delta virus is a circular single-stranded RNA virus, so that's the genome. And it encodes one protein, small delta antigen. So from this genome is made an mRNA. It's got a termination codon there, and that codes for small delta antigen. This allows the RNA to get packaged into virus particles. But a certain fraction of the time, that termination codon is edited by an RNA deaminase. So it removes amino groups from the bases. And in this case, it changes the A to an I, an inosine. When this is copied to the negative strand, the I templates for a C. So basically the I looks like a G. So instead of having a T there, the termination codon, it now has a UGG, which is a trip codon, and now you can make a longer protein, make large delta antigen. So that's an example of non-templated RNA synthesis or RNA editing. Now, in addition to mRNAs, cells encode lots of other RNAs. In fact, most human transcripts don't encode proteins, and they're classified into short and long bases, short RNAs and long RNAs, 200 being the cutoff. They have a variety of regulatory function, and they're encoded in viral genomes. They include microRNAs, long non-coding RNAs, circular RNAs, which have all sorts of functions that are just being explored and which we don't know about. Now, microRNAs, we know, regulate the existence of mRNAs. So microRNAs are made as precursors by Pol II. They are then chopped up into short pre-miRNAs. They're exported and associated with proteins called dicer. And eventually they are present as a single-stranded 21 base RNA, which can target RNAs that have the same sequence. So for example, they could bind to mRNAs and cause translational, not transitional, translational suppression. How many years has that been there? Oh my gosh. And they can also cause mRNA degradation. So these argonauts bound to the siRNAs have an endonuclease that can cut up. So these microRNAs are involved in gene regulation in the cell and also in virus-infected cells. An example of a microRNA having a function in a virus infection is miR-122. miR-122 is a liver-specific microRNA. It's involved in cholesterol metabolism. It's absolutely required for the replication of hepatitis C virus. Hep C is a hepatotropic virus. It doesn't infect any other tissue because no other tissue has miR-122. And so what miR-122 does, it binds to a sequence in the 5' end of the viral RNA. So there's the RNA genome. It's highly structured, and there are 2 miR-122 binding sites in the 5' end, and essentially stabilizes the genome in cells so that it can replicate. If you put miR-122 in another cell type, a non-hepatocyte, the virus will replicate in it. So it's essential for replication. And a company has made a complement of miR-122. It's called an entagomere, and people are doing this with other targets as well. And that inhibits virus replication. So if you bind up the miR-122, it inhibits virus replication. So it's a cool example of how a miR makes a virus liver specific. There are circular RNAs made also by back splicing. So instead of having 5' to 3' splicing, you go 3' to 5', and you make circles. Like here, you can make a circle of just 2, of 2 and 3, of just 3. And there are thousands of these in uninfected, virus-infected, and some cancers are full of these circular RNAs, and we don't know what they do. But one thing they do is to act as sponges. They can bind to microRNAs. They can also bind to RNA binding proteins and kind of act as a storage region for those elements until they're needed. A lot to learn about those yet. And finally, the last aspect of RNA biology I want to tell you is RNA methylation. And this is in particular N6 methylation of adenosines. So the base adenosine, the base A has an adenosine in it up here. And there are enzymes in the cell that will methylate this nitrogen. And so there are writers, the enzymes are called writers that put the methyl there. There are readers that see if there's a methyl there. And then there are erasers that take off the methyl, right? Very clever naming. Readers, writers, and erasers. So the reader would say, there's a methyl group here, let's do this. And so one example is in the hepatitis C virus genome replication, the viral RNAs associate with lipid droplets in the cell before they're packaged into virus particles. And if you have certain residues methylated, that inhibits incorporation of the genome into the virus particles. And so if you deplete the methyltransferase or the readers, you can increase reproduction of hep C. So this is kind of an antiviral effect of the host cell. It methylates the viral genome and inhibits its reproduction. So beyond translation and splicing, lots of different activities with mRNAs. Okay, so today we talked about DNA genome transcription, how to get mRNA made from all these different configurations. And next time we're going to talk about DNA replication. So we did transcription, we made one or more proteins, now we can do DNA replication. How does that happen? We're going to talk about that on Wednesday.